Alright, so we're going to talk about the vulva formation in C. elegans. Now, first of all, the vulva, it is formed during the larval stage, and there are 6 cells which are called the vulva precursor cells. And these 6 cells, they actually give rise to the formation of the vulva. So in short, they're called VPCs. Now, if we take a look at the C. elegans, the position of the cells, first we'll find the gonad. So this is the gonad, and just adjacent to the gonad we are going to get a cell which is called the anchor cell. So this cell is called the anchor cell, and apart from these two we have the 6 cells which are the VPCs. So these are the 6 VPCs that we already talked about. Now this anchor cell, this plays a very important role in formation of the vulva. So the anchor cell, this secretes a factor called Lin3, which is the master regulator for this vulva precursor cells. And this Lin3 is actually homologous to something in mammals which we already know as epidermal growth factor. So Lin3 is in C. elegans. When we come to a homologous protein in mammals, we have the epidermal growth factor, or EGF. So if this anchor cell is destroyed somehow, then this is going to lead to the vulva precursor cells to form the hypodermal cells. So hypodermal cells, this is the default fate. But when Lin3 is there, when the anchor cell is intact, this default fate is overridden and we get the vulva, or the cells that are forming the vulva. Alright, so we talked about the position. Now let's dig a little bit deeper with the VPCs and what are the different fates that are there for the VPCs. So normally the VPCs would have 3 different fates. Now if a cell is close, or the closest to the anchor cell, the VPC that is closest to the anchor cell, this is going to have a primary fate, and this is going to form the central vulva cell, or central cell. Right. So next, if a cell is closest to the cells which are closest to the central cells, so closest to the central cell, then they would have a secondary fate and they're going to form the lateral vulval cells, or lateral cells. Next, farthest from the APC, or farthest from the AC, farthest from the anchor cell, they are going to have a tertiary fate, and it's not receiving any Lin3 because it is farthest from the anchor cell, and hence this is going to form the hypodermal cells. So this is how the fate of the VPCs work. So this is the primary fate, which is the central cell. The secondary fate is the lateral cell, and the tertiary fate, which is the hypodermal cells. Now if we look a little bit deeper, and let's say this is the anchor cell, and we have the 6 cells. Now, the cell which is closest to the anchor cell, we call it P6P. And on the right, we have the P7P and P8P. So on the left of it, we have the P5P, P4P, and P3P. Now, the anchor cell, like I said, it is going to secrete Lin3. So it is going to secrete the paracrine factor Lin3. And P6P is going to receive the maximum amount of Lin3 and hence going to have the primary fate. The cells which are adjacent to the P6P, that is the P5P and the P7P, they're going to receive Lin3 but not as high as in concentration to that of P6P. So this is going to have the secondary fate, and the rest of the cells, they would receive like very less Lin3, I mean almost equal to nothing, and hence they're going to form the default fate, that is the hypodermal cells. So that's how the signaling works, and that's how the fates are decided. Now coming a little bit more to the signaling. So we have Lin3, and this Lin3 is received by a receptor which is there on this P6P, or the cell which is closest to the anchor cell. Let me draw the cell. Okay, so this receptor that is receiving the Lin3 is called Let23, and once it is received, it relays a signal and activates another protein called Let60. Now this Let60 has got different signaling pathways. The first thing that it does is it activates another factor called Let39, and Let39, what it does, it inhibits something which is called F1. Now, what happens when this F1 is inhibited? Normally, F1 would activate the genes that are responsible for forming the hypodermis, which was the default fate. Now, if we can, inhibit this F1, no genes are activated which are forming the hypodermis, and hence the fate remains that of the primary cell or the central cell. Now, Let60 can do other stuff as well. The next thing that it does is, since, like I said, Lin3 is homologous to EGF or epidermal growth factor, so this can promote cell growth through a pathway called the MAP kinase pathway. Right? So MAP kinase pathway, which leads to cell proliferation or cell growth. Another signaling that it does, another factor that is activated by Let60 is DSL. The DSL stands for delta serrate ligand. Now DSL is a protein that, or a factor that can be embedded onto the membrane of the cell. Now what happens when this is embedded? The cell that are adjacent to this central cell has got receptors for this DSL. Let me draw the cell. So this is the adjacent cell, and this receptor is called Lin12. So Lin12 is receiving DSL, and while it is receiving DSL, it relays a signal and starts transcribing a lot of miRNAs or microRNAs. Now these microRNAs are specific for the Let23 mRNA, and since Let23 is inhibited by these microRNAs, what happens? The protein is not formed because the transcription is inhibited. The protein is not formed. Lead-23 is not there on the membrane to receive Lin3, and hence this whole signaling pathway is stopped. No lead-16, no lead-39, no DSL, no MAP kinase pathway, and hence this is laterally inhibited. So the process is called lateral inhibition. And this is a classic example of the delta notch pathway, how delta notch pathway works. So what we saw Lin3 is the primary instigator of the pathways inside the primary cell, and the lateral inhibition is stopping the cells that are there on either side of the primary cell, and the lateral inhibition is stopping the expression of Lin3 or lead-23, and lead-23 is not being translated and thereby not receiving Lin3, thereby having a secondary fate. And when you talk about the tertiary fate, like the P3P or P4P or P8P, they do not receive Lin3, so we do not need to worry about all this lateral inhibition and stuff. So that's about it. That's how the signaling mechanism of Lin3 actually works, and how the vulval cells are formed, and what are the different fates of the vulva precursor cells. I'll see you in the next one. Bye-bye.